Ribosome. Ribosome is the another universal organelle. It is present in both the eukaryotic as well as the prokaryotic cell. Okay, and they are very tiny granule organelles, and they are found inside the cytosol, means in the cytoplasm, and sometime in the eukaryotic they are attached to some organelles. We'll discuss here only. Okay, so they play a very important role in the protein synthesis by translating the genetic information which is conveyed by the messenger mRNA, okay, messenger RNA that is called, and into the functional protein. Okay, so ribosome are basically composed of the two subunit. One is the larger subunit and the another one is the smaller subunit, and each of which is made up of protein and RNA molecule. So this one is very important question from the competitive competition, because ribosome, it is made up of protein as well as the RNA molecule. Okay, let's see what are the different characteristic or the features of this ribosome. So the ribosomes was first observed by the Palade in the 1953 under the electron microscope, and their nature and their function we all know. They are known as the protein factory. Why? Because they are responsible for the protein synthesis, and this process is known as the translation. See, the two different processes are there. Transcription is from DNA to the mRNA, that is known as transcription. But when the mRNA is converted, or they are responsible for the synthesis, for the generation of the protein, that is known as the translation. So protein synthesis, basically they translate the genetic information which is stored in the mRNA into the protein, and it also facilitates the productions from the amino acid. So the process, it occurs in the three different stages. The first one is the initiation, second is the elongation, and the third one is the termination. As the name suggests, the initiation means the beginning of the protein synthesis. Elongations, the amino acids, they are being added to the growing polypeptide chain, and the third one is the termination, that is the conclusions of the protein synthesis occur. Now, the moment of the protein, once the protein is formed, and after the protein formation in the ribosomes, the protein actually move to the different area of the cell for the various cellular functions. And they are of the cellular importance. Why? Because they are essential for some cellular functions, as they produce a protein which are crucial for the structure as well as for the function of the cell. That is why the ribosome is very crucial organelle, we can say, and it is single membranous, means or we can say no membranous. Okay, no membranous is the apt word. So it is because no membrane, while all other organelles such as endoplasmic reticulum, Golgi's, they all have the double membranous organelle, but the ribosome is a no membranous, or they do not have the membrane. So that is why it is known as the smallest as well as a non-membranous organelle. This is the structure of the ribosome, where we can see that the ribosome, basically they have the top two subunits which are joined to each other by interaction between the rRNAs in the one subunit and the proteins in the other subunit, and they are usually located in the cytosol, like in the prokaryotic also, because they do not have the endoplasmic reticulum. So obviously the ribosomes are scattered in the cytoplasms only. But in the eukaryotic organisms, the ribosomes are either present in the cytoplasm, just like in the prokaryotic cell, or they are attached to some organelle. Like in this diagram, the ribosomes, the green dot, they are attached to the endoplasmic reticulum. Okay, so they are either scattered in the cytoplasm or they are attached to some organelle such as the endoplasmic reticulum. So they are located inside the cytosol, and they are found in the plant cell as well as in the animal cell. Now, the structure, it is located in the ribosomes exist in the two form, and in the that is the free form, which is usually found in the cytoplasm, and the bound form is usually when they are present in the endoplasmic, or they are attached to the endoplasmic reticulum. Okay. And we know that when the ribosomes get attached to the endoplasmic reticulum, then this endoplasmic reticulum is known as the RER, that is rough endoplasmic reticulum, because of the surface become rough. That is what is known as a rough endoplasmic reticulum. Okay, the bound and the free ribosome, they both are similar in the structure, and they both are involved in the protein synthesis. However, the synthesis of the protein may be different, because the ribosome which are synthesized in the endoplasmic reticulum are of the secretory nature, because they are synthesizing there, but after the formation of the protein, they are being sent to the Golgi apparatus. From the Golgi apparatus, they are sending to the plasma membrane, and then if they are destined to go outside, then they are moving, or they are excreted out of the cell, or they are destined to some destination, or the targeted organelle, then they may be sent back to the destined organelles. So this is all about the ribosome structure. Next, we will discuss about the subunits of the ribosome. So the ribosome basically consists of the two subunit, the small subunit and the large subunit. And these two subunit, they work together during the process of the protein synthesis. Like they are looking here as a separate unit, but when the protein is synthesized, like in the next diagram, we can see that both the units work in the coordinated manner when the protein synthesis actually taking place. So the ribosome consists of the RNA as well as the protein part. The RNA component is known as the rRNA. Okay, and the protein component consists of the various ribosomal protein. Okay, the smaller subunit, basically what actually it do, the smaller subunit, it read the genetic information and thus bind to the mRNA. As we can see here, the ribosome is smaller subunit, it binds to the mRNA. Okay, while the larger subunit, it catalyzes the peptide bond formation and binds to the amino as acylated tRNA. And we can see here the tRNA